So good evening, everyone, and welcome to this ROS-1 international meeting. So my name is Alfonso Aguarón, and I will be your host today. And as we have a very tight agenda, I will give a very brief welcome and introduction to all of you. So first of all, I would like to mention that this session is possible thanks to the Spanish group for people living with lung cancer, who have been the main organizers. And it has also been co-hosted by Lung Cancer Europe. Before we get started, I would like to thank our speakers here today. I know your agenda is very tight, and we really value your precious time today. So I would also like to thank the supporters of this meeting. So for this meeting has been a collaboration of some sponsors, Pfizer, Takeda, and ROS. It would haven't been possible without their contributions. So thank you so much for helping the Spanish group of people living with lung cancer to organize this session. So without further ado, I would like to explain you a little bit about the dynamics of this session. If you could have the chance to look at the agenda, we have a pretty interesting and very comprehensive program today. So we'll be having our speakers touching base on different topics related with ROS-1, and we will have some time for questions at the end of the meeting. So if you would like to have your questions asked to the doctors, I kindly ask you to use the options that the platform is providing you to send your questions in writing. I will pass them to the audience. Also, you have the chance to enable the simultaneous translation. The full meeting is going to be streamed in English, but you will have the chance to listen into Spanish just by selecting the language on the platform you just entered to follow this session. So without any further ado, I would like to give the floor to Dr. Enriqueta Felip, head of the Thoracic and Head and Neck Cancer Unit at the Vall Institute of Oncology in Barcelona. So she's going to speak us about the ROS-1 biomarker diagnosis method. So Dr. Felip, thank you very much for your time, and the floor is yours.
Thank you, Alfonso. And also I would like to thank Bernard Gaspar for the invitation. It's a real honor for me to participate in this meeting. So I'm going to review ROS biomarker diagnosis methods. My disclosures. So today we are going to discuss ROS. ROS is a gene located in chromosome 6. ROS encodes a tyrosine kinase receptor and was originally described in glioblastomas. ROS-1 was first described in non-small cell lung cancer in 2012, and there are a number of partners with ROS. We know that ROS and ALK share many features. Even from a clinical point of view, ROS-1 patients and ALK positive patients have majority adenocarcinoma histology, have a younger age at diagnosis, and also is a group of never or light smokers. And today we are going to discuss the methods for ROS-1 detection, and I would like to highlight three methods. FISH is the method required in Profile 1001 trial. We have also immunohistochemistry, the D4D6 monoclonal antibody, and also NGS. Probably the preference depends on availability and also experience, but in all cases, quality control is mandatory. As I mentioned, in Profile 1001, that was the study that was the reason for the approval of crizotinib in ROS-1 positive patients. FISH was required for study entry. And as you know, with this technology, the response rate with crizotinib was 72%, and the median progression-free survival was 19 months. There is one paper analyzing potential algorithms for testing ROS-1. And according to this paper by Bubendorf, there is a possibility to pre-screen using immunohistochemistry, but for those patients or for those tumors ROS1 positive by immunohistochemistry, there is a need of reconfirmation by a second method, FISH or NGS. So I would like to mention also a Spanish paper published in JTO analyzing a new immunohistochemistry clone, the SP384. In this study, a total of 43 ROS1 FISH positive tumors and also 193 ROS1 FISH negative samples were analyzed using two antibodies, the D4, D6, and also the clone SP384. And the conclusion was that this new antibody, the SP384 immunohistochemistry clone, has also an excellent sensitivity for determining ROS1 tumors. In ROS, we know also the mechanisms of acquired resistance after crizotinib. In this paper in the New England Journal of Medicine, it was described the G2032R mutation as a mechanism of acquired resistance to crizotinib. And also this was also published by Gaynor analyzing 16 tumors from patients receiving crizotinib and then progress. And in these tumors, resistant mutations were seen in 53 of specimens, and the G2032R was seen in 41% of tumor samples. And today, in general, in lung cancer, we have the debate, is better to perform a single gene testing, or do we prefer NGS, next generation sequencing technologies in tumors with non-squamous histology? So according to the ESMO guidelines, if available, multiplex platforms for molecular testing are preferable, probably very similar from the American group. The multiplex genetic sequencing panels are preferred where available over single agents, single genes, to identify other treatment options beyond EGFR, ALK, BRAF, and ROS. And also the Spanish guidelines are going in the same direction. So we have to consider NGS platforms for the testing of stage 4 non-squamous histology. And I would like to highlight a very important paper from ESMO analyzing this is by the ESCAT group. And in this paper, the authors recommend the use of NGS testing for all non-squamous tumors in patients with a stage 4 disease in order to detect the level 1 alterations. And among them, you can see ROS1. So this is again from this paper from the ESMO recommendations for NGS in advanced non-squamous histology. As I mentioned, there is a recommendation to perform NGS in this situation, and the level 1 alterations include EGFR mutation, ALK, MET-exon 14 skipping mutation, BRAF V600, ROS, NTRK, and RET. There is a need also for liquid biopsy in non-small cell lung cancer, and again, in a number of papers, we have seen that ROS may well be determined in some liquid biopsy determination. It's true that a positive finding on plasma can guide the treatment, but a negative finding may still require testing. But liquid biopsy is really important in non-small cell lung cancer, and there are other potential advantages. Among them, the turnaround time that is shorter with plasma NGS determination when compared to tissue NGS determination. And again, I'm going to refer to these NGS ESMO recommendations published in Annals of Oncology. In Advanced Non-Squamous Histology, it is recommended that tumor or plasma sample will be analyzed using NGS technology in order to detect level 1 alterations. But there is also the sentence mentioned that considering the high frequency of fusions or rearrangements, RNA-based NGS or a DNA-based NGS designed to capture such fusions are the preferred options. So let me just finish by saying that in lung adenocarcinoma, genotyping increases the chance for overall survival improvement. So there is a need to genotype all non-squamous non-small cell lung cancer tumors, and also probably some squamous cell carcinoma tumors in never smokers. ROS-1 must be determined, and the recommendations also is to perform a next generation sequencing panel, including not only ROS-1, but also EGFR, BRAF, MET, ALK, RET, and TRAC. And also is also important to remember that we need liquid biopsy in our clinical practice. And finally, it's important also the collaboration among all the team, and you have started all the molecular tumor boards in each institutions in order to discuss, to find available treatment options for those patients in which we have to determine NGS and we have molecular alterations. So thank you, and happy to discuss later on in the discussion.
Thank you so much, Dr. Felip, for this introduction to the diagnosis methods. Before going to our next speaker, I would like to remind you that you can send your questions just by typing them in the platform that you just joined, right below the video where you can see the speakers and the slides. You can send your questions because we will have time to answer them at the end of the session. So I'm pleased to welcome our next speaker, Dr. Benjamin Besse. Thank you very much for joining us today. So Dr. Besse is a professor of medical oncology at the Paris-Saclay University, and also full cancer specialist at the Gustave Roussy Cancer Campus in Villejuif, France. I hope my French is not too bad, Dr. Besse. So Dr. Besse is going to talk us about the treatment for ROS1 lung cancer. So without further ado, Dr. Besse, the floor is yours. Thank you.
Hello, and thank you very much for this invitation. It's a great honor to be there and to speak to patients. We speak to patients during consultation, but explain the disease, explain the treatment. It is always something that is very good. These are my disclosures. So who are you? Who are the patients with ROS1 and LCLC patients? Well, first of all, it's a rare disease. It's a rare disease because it's roughly 1% of all non-small cell lung cancer. So it's a tiny proportion of the patients. It means that it's only one patient out of 100. It means that you have to screen a lot of patients to find one patient with this type of cancer, but it's really, really important. But we have a way to have a previous idea who could be more prone to have this molecular alteration. Most of all, lots of these patients are either never smoker, either light smoker that have smoked, for example, less than 10 years during their lives. They are younger than the others, in particular than the cancer we find in smokers. The histologic type is, in general, adenocarcinoma. In squamous cell carcinoma, a different type of cancer, we hardly find them. And they can have some brain lesions. Risk factors, it's the risk factors of lung cancer is usually the smoking status, the tobacco. But for ROS1 positive NLCLC, it's a bit different. It's mostly found in never smoker or light smoker. So what is the cause of this cancer? Well, the radon is thought to be the second cause of lung cancer. There is a strong awareness of radon in the US because when you sell your house, you have to test the quantity of radon to be able to sell the house. And 10% of the houses in the US have a level of radon that is higher than the one that the regulator asks. And in Europe, it's the same, but there is less awareness of radon. Radon is a radioactive gas that you find in some region where you have a lot of volcano, for example, and it's a natural gas in the air, but it accumulates inside the house. So you need to have a specific ventilation system in the house or to open the windows to decrease the level of radon. At EORTC, an European organization, we will launch together with Laura Mesquita a European study to evaluate more specifically the risk of radon in ROS1 positive NLCLC. Well, ROS1 is a, let's say, a young entity among lung cancer. It has been described a bit more than around 15 years ago, but in fact, the real entity of ROS1 positive emerged in 2012 when we got treatment for this entity. Since then, it's a very well-known entity. So in non-small cell lung cancer, there are three big groups of stages. A third, roughly, of the patients will have what we call an early or localized stage. Usually, these patients are treated by surgery. They could have some post-operative chemotherapy sometimes, but for the ROS1 positive patients today, we don't offer any specific treatment. It's, as for the other type of cancer, surgery and chemotherapy. Sometimes when the patients have impairment of the lung, it will not be surgery, but it will be radiotherapy. The second group is what we call the locally advanced. The disease is still in the thorax, but it's too big to be candidate for surgery. So the definitive treatment will be, in general, radiotherapy. We can add, on the top of radiotherapy, chemotherapy, and sometimes this lesion can be resected. But for the patients with ROS1 positive NLCLC, we don't offer additional treatment. Finally, the patients can be diagnosed at a metastatic stage. It means that the disease has spread outside of the primary site, either in the other lung, either outside the thorax. It's in these cases that the ROS1 inhibitors has been developed, and this is what I will address in this presentation. So first of all, what is striking for us, the physicians, the lung cancer physician, is that the ROS1 positive NLCLC patients live much longer than the other patients with metastatic disease. We think that it's because we have the good target, that is ROS1, and the good treatments. We can do something similar in ALK positive NLCLC. It's a different kind of cancer, but it has some similarity. And there again, we have the target identified and good drugs so that we can prolong the life of these patients. So we talk about a lot of overall survival. We talk a lot about progression-free survival, meaning the time during which the patient will receive the treatment. It's really the way we talk between physicians, but when we talk to patients, we should always remember that below the curve, there are hundreds of patients living because of the drug. So it's true that you see this curves as an individual, when we see these curves as a group of patients, we might have different views here, but we have the same goal. It's to treat and delay the progression of the disease as long as we can. Crizotinib is the first ROS-1 inhibitor that was tested in lung cancer patients. In the first study, we saw that the response rate, as we say, was 72%. What does it mean? Well, look at the left part of this slide. You will see that each bar represents one patient treated with crizotinib. 53 patients were treated. Each time the bar goes down, it means that the lesions, the cancer of the patients, decrease during crizotinib treatment. As you can see, only 2 patients had a primary resistance, meaning that the disease increased during crizotinib. It's a very impressive results. And when the lesion decreased by at least 30%, we call this a partial response. This is what we capture in the term overall response. This is something I will refer to during this presentation. In Asian patients, we saw exactly the same figure. The PFS, meaning the median time where the disease is controlled by crizotinib, ranged from 15.9 months to 19.3 months. What does it mean? It means, for example, that half of the patients will have a disease progression after 9.3 months of treatment. For us, for lung cancer physicians, it's a very long time under treatment. A lot of additional small studies have shown that the role of crizotinib is major and that this is a very, very potent drug for ROS1 positive non-small cell lung cancer. As you can see, the number, it's called N, it's the number of patients in each cohort, is quite small, but still it gives an impressive results. There are other ROS1 inhibitors, ceritinib, for example, cabozantinib, entrectinib, repotrectinib. Not all these drugs are approved for ROS1 and LCLC. Crizotinib, entrectinib are today approved. The others are not approved yet. But it's just to give you a glimpse that all these drugs that inhibit ROS1 might have very different level of toxicity. If you look, for example, at the green bar, it's the nausea. You can see that it's very different among the ROS1 inhibitor. There are no perfect ROS1 inhibitor, unfortunately, but it's a balance, and we try to select the one that will be the best for the patient. There are, as I said, other ROS1 inhibitors that are also very potent, ceritinib, entrectinib, lorlatinib. This is the response rate in the patients that never received any treatment for their ROS1 positive and LCLC. And as you can see, the PFS, meaning the median duration of control of the disease, is very good with all these drugs. While at one point, the patients can see his disease grow, although he's still taking the drug. While we did a very interesting study, we took some blood in patients that were still on treatment for at least 3 months. What we saw is that in half of the patients, the quantity of the drug in the blood was not as high as expected. And in fact, it was below the level expected. Why? Maybe because some patients do not take their pills exactly the way they should, and maybe because there are interaction with other drug or with natural product that they can take. And it has been shown that if you take a population that is exposed to crizotinib, this is 2 different curves of the median PFS, the median control of the disease. And as you can see, the patient that had a higher dose in the blood seems to have a higher duration of control. This is something that we don't use so much, but all the patients take the same dose of the drug, and maybe in the future, we will have to adapt this. But in general, when the disease grows, it's because a new mutation has appeared inside the ROS1 gene. It modified the ROS1 protein, and then in this little pocket of the ROS1 protein, where the ROS1 inhibitors can block the ROS1 signal, because of this mutation, there is a new let's say a new shape of this pocket, and sometimes the drug cannot enter in the pocket anymore. So additional drugs have been designed. We call them next generation inhibitor. Repotrectinib is one of these drug. It's active against a mutation that we call G2032R. It's a complicated name, but just to say that when you find this resistance mutation, most of the inhibitor will not work, but repotrectinib. If you give this drug to patients already exposed to crizotinib, you see a very nice response, and it's the same with lorlatinib. If you give this drug directly to patients that never receive treatment, it seems also to be a good solution. The patients that have ROS1 positive and LCLC frequently have brain metastases, and these brain metastases also respond to the ROS1 inhibitor. This is a good news. So overall, what is the best sequence? Well, we don't know. Should we give the stronger drug first, or should we start with the older drug, crizotinib, and then at the time of progression, switch to a stronger drug? Well, today we don't know. We don't know if it's better to start with crizotinib and then give a second drug, or to start with a stronger drug first. Finally, I will just end with this. What are my issues as a clinician? Well, I had an issue with crizotinib, for example, in my country, in France, because what we call the HAS, which is the regulator that decides if the drug will be refunded or not. It's the Europe that decides if the drug is approved, EMEA, but then at each country level in Europe, each country decides if we'll refund the drug. Crizotinib is very potent in ROS1. We have very strong data. And still, my country decided that the benefit of crizotinib was really minor, that there was no strong data, and that we should randomize the patients from crizotinib to chemotherapy. So the patients that were never treated should be randomized between crizotinib and chemotherapy when we see that crizotinib has a very, very potent drug. And they have decided to refund the drug only second line. They say, well, we don't have enough data, start with a chemo, and when the chemo failed, give crizotinib. I'm completely against that. It's really not a good decision. And I prescribe crizotinib as a frontline drug. And I think that there is a gap here between what we know, what you feel, and what some states decide. We don't have randomized data, but we have retrospective data from collection that show that the patient with crizotinib had a much better outcome than the patients treated with chemotherapy. So in my country, crizotinib was only refunded 1 year ago for the patient with ROS1 and LCLC. It was much too late, much too late. Lorlatinib, that is also a potent drug, is not available for ROS1 positive and LCLC, although there are some clinical trials now. But we need to fight again, and we need to find better way, at least in my country, to fight physicians and patients to really get access to the drug. Dr. Ramon from Barcelona gave me this drug that really nicely show what are the issues in the access of the treatment. From the right to the left, you have the time where the drug access to the market, and then you have the percentage of patients that had access to this drug. And as you can see, there are a country where the drug is available very, very, very late. All this group of cancer patients might help to speed up this process. So my conclusion is that ROS1 and LCLC is a rare disease. We have now 2 generations of ROS1 inhibitor, but only 1 generation is available to be prescribed today, crizotinib and entrectinib, that has a good activity. Repotrectinib seems to be the more potent, but we have to see more mature data. We should better understand the mechanism of resistance, but today it's not standard of care to rebiopsy the patients. It's not needed. Chemotherapy is still a good option when the patients fail the TKI, and immunotherapy is not there yet. As physicians, we need to access large screening to find the patients with ROS1 positive and LCLC. As patients, you need access to the drug, and we should fight together.
Thank you so much, Dr. Besse, for this challenging presenting all these treatments in such a short period of time. I know that's challenging. I also take the chance to adapt to that message that we should really tackle together these access issues. Sadly, this is something that is not exclusive to France, but these inequities in terms of access to treatment and diagnosis seems to be a major problem in most regions in Europe. So that's a good message, and I'm pretty sure that we'll have time to discuss around it at the Q&A part. So thanks a lot, and right now it is my pleasure to introduce you to Dr. Patrick Pauwels. He's the head of clinical pathological anatomy department at the Antwerp University Hospital. So thank you very much, Dr. Pauwels, for making your time to be here today. And Dr. Pauwels is going to talk us a little bit more about the research line in ROS1 biomarkers. So without further ado, I will share the screen, and the floor is yours, Dr. Pauwels. Just a second. Here it goes.
So first of all, thank you for inviting me. It's a real pleasure. I have to speak about ROS1 biomarker research lines. I will stick to the research lines and will end with something about the biomarker. Next slide, please. So research in ROS1 fusion positive lung cancer is going on, of course. And one of the most challenging research topics we have today, as has been said already, is the problem of resistant mutations. Next. But how can we look at ROS1 fusions and resistant mutations in a research setting? One of the first things we use traditionally is cell lines, but there I have to tell you something. You know that probably that book about the immortal life of Henrietta Lacks. This was a woman who died a long time ago of cervical cancer, but her cervical cancer cells survived, were immortal. And a lot of experiments have been done on these cell lines of cervical cancer. But the problem we face is that these are cervical cancer cell lines. And with molecular techniques, we can introduce ROS1 fusions in these cells, but we are still speaking about cervical cancer cells. Of course, we can use other cell lines, fibroblasts and whatever, and introduce ROS1 fusions, but still the problem remains that these are not lung cancer cell lines. Next slide. Next slide. You went back. Next. Yes. But fortunately for us, we are very happy that there was something called as a ROS1 cancer model project, and these cell lines are now derived from patient samples, and they have indeed ROS-1 fusion. So you see ROS-1 fusions in lung cancer, it's rare, and we have only these 7 cell lines, of which only 1 was a cell line resistant to crizotinib, that we can use for experiments of how to conquer these resistant mutations. Next. Fortunately for us, molecular biology gives us a tool for helping us out of this problem. With a CRISPR-Cas9 technique, you can delete, remove part of a gene very selectively, and the gene, the DNA does not like that, will want to repair this, the situation by using non-homologous end-junction repair. By this way, you can remove part of ROS1 gene and introduce a DNA sequence that you want to examine. So when we are using ROS1 fusion positive cell lines, by this CRISPR technique, we can introduce these resistant mutations. So we are not talking about fibroblast, cervical cancer cell lines, but of the real ROS1 fusion cell lines. Next. What we do then next is, of course, we will start to select all kinds of drugs that are tackling ROS1 and see what happens. Which mutations will we introduce in our ROS1 fusion cell lines? Of course, the mutations that have been proven to give resistance to crizotinib and lorlatinib in the patients. So, for example, in crizotinib, we have already 7 mutations described as giving resistance. So in these cell lines we have now, we will introduce these mutations, and we can experiment with several drugs to tackle these resistant mutations. So cell lines are good, but we are better and better by having cell lines from real ROS1 fusion positive tumors. Next. I think that you all have seen such slides with 3-dimensional figures about, for example, ROS1 or whatever molecule. There we enter the topic of structural biology. By crystallography and other means, you can have a 3-dimensional insight in a ROS1 molecule. And you can also see what happens if you introduce in your computer model the mutation you know. You can see what happens in the 3-dimensional structure of your ROS1, and you can see how it will affect the binding capacity of your medication you will give. It sounds a bit futuristic, but next slide will show you an example of an ALK positive case. This is really a famous case report of Alice Shaw. And what happens is, next slide, that you had an ALK positive patient, was on crizotinib, developed resistance, some more treatments, then lorlatinib became available, was introduced, but there was resistance to lorlatinib, and at the end you see again crizotinib. Why again introducing crizotinib? Next slide. Well, what happens is that the patient was resistant to crizotinib due to one mutation. This mutation stayed. Then the patient came resistant to lorlatinib due to another mutation. And by looking at the three-dimensional structure of this ALK molecule and introducing these two mutations, the computer predicted that you could, by the change in the 3 dimensions, again use crizotinib, and with a good result. So structural biology will inform us also about the possibilities for the new drugs. Next slide. Now, we all know these figures, EGFR, all these pathways, and it looks very easygoing, very understandable. But the reality you see in the next slide, which I always use when I start my lessons on the molecular pathology. What you see there is in fact the EGFR pathway. And you see a lot of things that are happening together, a lot of pathways that are involved. And there we come to the chapter of bioinformatics. Next slide. So what's bioinformatics? Well, the beauty of bioinformatics is that you collect a lot of data, a lot of input, then the magic of bioinformatics will work and will give you a meaningful output. Next slide. Now, as bioinformatics, what would you like to introduce in your system, in your computer? Well, for example, of course, the mutation, but also maybe the concurrent mutations that are always there. It's not only ROS-1 fusion that happens, there are always concurrent mutations. You can also introduce the data of your epigenetics. You can also introduce data of your proteomics. You can introduce data of metabolomics, and probably all other kind of data. And so the computer, bioinformatics, artificial intelligence will give you an algorithm. And I think that the next step in the biomarker study will be this, that we will have algorithms that will predict success of therapy and even predict what we should use as therapy. And that with this system of gathering data and making new algorithms, that we are going to a new kind of precision medicine, meaning that we will have really the tool for personalized medicine. Next slide. Thank you for your attention.
Thank you so much, Dr. Pauwels. It really looks like this kind of magic, to call it somehow, which sounds like science fiction. The good thing is at the end that thanks to science and research, we can see that there are very promising things going on for the ROS-1 patients, and this brings certainly a lot of hope for many patients. And I really think that our audience will really appreciate to know about that. We'll have the chance to speak more in detail about these research lines in our Q&A session. Just to remind you, to our audience, if you just joined shortly, you can send your questions to the speakers, because we will have a question and answer session at the end of the panel. So, my pleasure now to introduce you to our next speaker, Dr. Antoine van der Wekken, pulmonologist in the Department of Lung Diseases and Tuberculosis at the University Medical Center of Groningen in the Netherlands. Thank you so much. I know your agenda, Dr. Van der Werken, like every one of us, is very tight today. So thank you so much for being on Friday with us. And Dr. Van der Werken is gonna talk us and drive us around the work that has been developed in Europe in ROS-1 research compared to the work done in other countries. So Dr. Van der Werken, the floor is yours.
Okay, I would like to start with thanking the organizers to introduce me and to give me the opportunity to give a talk. So I've been assigned to talk about the work developed in Europe and comparing that with other countries for this ROS-1 semester. I first wanted to start with actually thanking the ROS-1ers, because I think that it's one of the major platforms that has made a lot of things possible in the things we learned from ROS-1, and that sharing knowledge among a lot of patients who are still having a rare disease helps us in well, knowing more about it. I would like to talk about a couple of things. I think the major items I want to tell you is that we have to look at cancer care and as well at cancer research. And the three items I will discuss in the couple of minutes is about what can we learn from example from the USA, what do we know, what's happening in the European Union, and the fact that we are working on a collaboration in Groningen and how we can use that. So first of all, what can we learn from the USA? I think that there are a couple of very large centers in the USA, and that they treat a lot of patients with rare cancers, and also for ROS-1, for example. And because they all speak the same language, it's more easy to travel for a longer time and just still be able to speak in your native language. On the other hand, there has been made a collaboration of different centers who joined to learn more about ROS-1, and they have also made some papers about what do we know about ROS-1. And as Professor Pauwels and also Professor Besse already told, there are differences in ROS-1. So, for example, at the left upper side of the slides, you can see that there are different fusion partners, and that have been found in, well, the patients they have tested at these centers, and for example, Colorado University. They also learned that there are different mechanisms of resistance, and that can be, as Professor Paul already mentioned, in the same gene, so in ROS-1. So there can be mutations there. But there are also a lot of items we don't know. So as you see at the right bottom, that there is no resistance mechanism known. And of course, a lot of research, what you see at the right side, has been performed to know what to do when you have a new mutation in ROS-1, to know if there is a possibility of combining drugs. That's what they have done already in the collaboration of the USA. The other problem for the EU actually is that we have many different countries. There are many different ways of treating. So, for example, in the Netherlands, the lung tumors are treated by pulmonologists, and there are some other countries where pulmonologists do the treatment, but in most other countries, it's the medical oncologist. So there is a difference in who does it. And the other hand is that in some countries there is centralization of care. So you see that patients do go to a center who has more knowledge on rare cancers like ROS-1, and other countries it doesn't work like that. So I think based on what I've seen, that it's good to have centralization of care, of course. And the other thing is that it's the way to go to have newer studies, to have the possibility to get newer drugs when patients get resistant to for example, crizotinib. But the question is, how should we do that? Should we use all kinds of things we know? Well, I will show you how we do it in the Netherlands. In the Netherlands, there are many hospitals who do lung cancer care, but we now have said that every patient with lung cancer should at least be tested for a couple of genes. So, for example, EGFR and KRAS, and also ROS-1. So we try now to make sure that every patient in the Netherlands will be tested for ROS-1. And if there is a ROS-1 found, for example, or an ALK or a RET, then the patient should be sent to one of the 6 centers I have shown you in this slide. So Groningen is in the right upper part. It's in the northern part of Netherlands. That's where I live. But, well, there are around the Netherlands a couple of large centers who have the best knowledge on treatment of ROS1. They know the side effects. There is the knowledge about new treatment options. And in some centers, we do have the new treatment options with, for example, repotrectinib. And we're working also on newer treatment options, of course. Well, these are the centers where also the research is performed. And I think that Umcg is one of the centers where we try to get more knowledge on ROS1. But can we share that knowledge, and should we collaborate with other centers in Europe? How should we do it? It's quite hard to know. So, for example, we can maybe use the clinical cancer centers in Europe. You see here a picture of all the centers which are certified or in the A&D process. So Groningen is not on this map, but we are in the process. So there should be a star at the arrow. But you see that, for example, in Germany, there are no centers. It doesn't say that the care is not good at all, well, because I think it is. So I think that's not the way to go. But still, we have to think about centralization of care in every country, and every country has to do it by themselves to make sure that will happen. And I think that's very hard to make that happen. And to go on research, how should we go there? There are some platforms like the Europe PDX platforms where many centers are involved. Maybe that's a platform we could use to do ROS1 research as well. But it's still hard to get real in contact and make that happen. So that was the reason Merel, which is one of the ROS1ers, asked me and also another university in Groningen to make a collaboration to perform more ROS1 research. And that's what we have been working on the past couple of years. So together with Stichting Merels Wereld and the Hanse University of Applied Sciences, we are now performing some ROS1 research. So at the Hanse University, one of the PhD students is working on different cell lines. So here you see an example of the cell line she is working on, which has a ROS1 fusion with a GR mutation. It's still a BAF3 cell line, so that means it's not a real lung cancer cell line, as Professor Pauwels already mentioned. But I think these are little steps to go forward, and we try to find out whether we can introduce new mutations in the tumor cells and find out which kind of drugs will help. And I think that work is done in many centers, but still we have to use it. And the thing is that we can use patient information to introduce in these kind of cell lines. But the other problem is that there are also other mechanisms. So we try to get patient tissue to make cultures, to get resistant cultures on TKI. So at the left bottom, we used resistant cultures where we have mutations in ROS1. We compare it to the 3D modeling, as Professor Pauwels already showed you. We try to do a little bit the same and compare whether different TKI can work in those mutant cell lines. But on the other hand, we have cultures where there is no mutation found. We don't know what the resistance mechanism is, so we need to dig in deeper and know which kind of mutation that will be. So then we have to do mutation analysis using exome sequencing or RNA sequencing and so on. And these are the items I think we need many patients and patient material to be involved in this research. And we also need to collaborate with other centers other than only Groningen. Well, at the Umcg, these are one of the first results we had. So what we do, we get a biopsy or pleural fluid from the patient who has resistance on, for example, crizotinib or lorlatinib. We make a single cell suspension, and then we try to get a low passage cell line, and then we do the sequencing and also some drug testing. And you see at the bottom figure that we have the growing of the tumor cells in a flask, and that's been done by Fenneke Swieringa and Wei-Ting Lee, two of my PhD students. And we hope to get more tissue, of course, and that's why we had made this collaboration with also Merels Wereld to involve patients and try to involve patients of whole Europe to see whether all these persons can be involved in helping their own disease to go, well, to know more about it. So next thing is I want to share a short video with you where we will let you see what we're trying to do in the collaboration. So I'll stop this one and I go to this one and share it. It takes about 3 minutes, so.
You have been diagnosed with a rare form of lung cancer, the ROS-1 fusion. The University Medical Center in Groningen, in other words, the UMCG, and the Hanse University of Applied Sciences, in partnership with the Merels Wereld Foundation, are carrying out scientific research into this ROS-1 fusion. As part of this study, the researchers are studying, among other things, the effects of different drugs on this form of cancer, for which they require tissue or lung fluid of as many people with the ROS-1 fusion lung cancer as possible. You can play an important part in this. You can do this by taking part in the scientific research. Taking part in this research is voluntary and does not cost you anything. If your lung cancer drugs no longer work as well, or if the lung cancer has grown, your doctor will take a sample of tissue or lung fluid during a puncture procedure in your hospital. If you decide to participate in the scientific research, during this puncture procedure, your doctor will take an extra sample of tissue or lung fluid. The researchers will use this extra tissue or lung fluid for the scientific research. It is important that you register yourself with the UMCG as soon as you know the date of the puncture. You do this by sending an email including your name, telephone number, and the date of the puncture. The UMCG will contact you and your doctor to discuss everything. In addition, please inform your doctor of your participation. After you have registered yourself, the UMCG will send you a special kit. The kit consists of a polystyrene box, including a small tube with cooling elements. The tissue or the fluid can be placed in the tube. When a tissue sample is taken, the tube will be half filled with a pink additive solution. When only a sample of lung fluid is taken, the tube will be empty. Be sure to follow the storage instructions. The cooling elements will need to be stored in the freezer until the puncture. The tube containing the additive solution will need to be stored in the fridge. The consent form is included with the kit. You will need to sign and include this when returning the kit. You take the complete kit with you when you go for your puncture in your hospital. Please ensure that after the puncture, the sample of tissue or fluid is correctly packed and cooled. You will receive instructions on how to do this. The kit will then be returned to the UMCG. The UMCG will send a courier. Thank you in advance for your cooperation. This research has been partly made possible by the University Medical Center in Groningen, Merels Wereld Foundation, and the Hanse University of Applied Sciences.
So in conclusion, I think we should centralize care for rare lung cancers, for example, ROS1, and I think we should try to collaborate, all of us, to get better insight in ROS1, also in Europe. So I hope patients who have lung cancer with the ROS1 hope to join in this project, and I also try to collaborate with other centers. So I already have spoken with people from Antwerp, for example, but we will try to get more knowledge with this start kick, actually. So thank you very much, and happy to take questions at the end.
Thank you so much, Dr. Van der Wekken, for your presentation, for showing us this project. And I think there's a good learning out there that despite the efforts made in Europe, I think with ROS1, the collaborative approach is certainly needed in order to get the better outcomes in research and at the end of the day to improve people's lives. I think this is a lesson that we have learned from the COVID-19 pandemic, that there is a way to really join the force together to speed up things, because at the end, this is a win-win for absolutely everyone in the field. So thank you so much, and there will be certainly some time to discuss this deeply in the Q&A session. So right now I'm going to introduce our next speaker, Dr. Santiago Viteri, lead clinician at the Medical Oncology Department of the Oncology Institute, Dr. Roussy in Barcelona. Thank you so much, Dr. Viteri, for being with us today. Dr. Viteri is going to talk us about the specific needs of ROS1 patients. So without further ado, Dr. Viteri, if you share your screen, the floor is yours. Thank you.
Thank you very much. I would like to thank the organizers for this meeting, for inviting me to share with you knowledge, passion, friendship, and just to create a community where we can all work together and care for each other, no? Which is in the end one of the main basis of our work as doctors, but also of our characteristics as humans, no? So I would like to say that this meeting is unique. It's not so common. We usually do a lot of meetings where we speak with other scientists or other doctors. We teach ourselves, we share our knowledge. But it is not so common that we do this sharing our knowledge to patients. And also the topic of my speech, specific needs of ROS1 patients, is also not so frequent. But I was wondering, why is this not so frequent if the principal players of this story are the patients? So we need to change this. And I'm happy that this session is a big step on this direction. So specific needs of ROS1 patients. I know I'm the last one to talk of the doctors. I know that they have. I was thinking, was guessing that they would review many of the main facts of ROS1 treatments, diagnosis, resistance, etc. So I tried to make a different approach, and I was thinking to myself, with the things I know as an oncologist, what will be my concerns, and if I was the patient. Okay, so I'll try to do this exercise, and I will show you what I was able to concoct. First of all, if I am a ROS1 positive patient, I need to be diagnosed correctly. As it has already been mentioned, this is a rare disease. It's infrequent. If we speak about non-small cell lung cancer, it's only about 1%. Some oncologists will not see ROS1 patients, maybe never, or until now. Some of them have never seen ROS1 patients. That's true. I'm dedicated to non-small cell lung cancer, and for years I was pissed off because I've never seen ROS1 patients for some years. But what is the issue on this? First of all, I would like to stress that ROS1 is not only a problem for non-small cell lung cancer. There are other forms of cancer that are driven by ROS1 fusions. Some of them are similar to the ones we know in non-small cell lung cancer, but some of them are specific for the different tumor types. As you can see, 0.5% of gliomas, tumors in the brain, some from adults or pediatric gliomas, can have ROS1 fusions. Spitzoid neoplasms that are a particular type of skin tumors are frequent in ROS1 fusions, and also a very specific tumor that is called EMT, myofibroblastic tumor, inflammatory myofibroblastic tumor, has around 10% of ROS fusions, but also has ALK fusions. So in this case, it's similar to non-small cell lung cancer. And in the other column, you can see other tumors that where reports of fusions have been published, but we don't know the real frequency. So one thing I was considering is that if ROS1 fusion tumors are infrequent, maybe we could join forces, not only non-small cell lung cancer, which is the most frequent of all these type of tumors, but probably in the biology, some aspects will be similar, and we can learn many important points from what happens in other tumors, and also other tumors could benefit from what the lessons we already have from non-small cell lung cancer, because it is only for ROS1 positive non-small cell lung cancer that we have approved treatments. For all the other tumors, we don't have approved treatments yet. And this was also been discussed before, but it is very important that we can detect the ROS1 patients. Otherwise, we will miss the opportunity to offer the best treatment. If we don't do the right diagnosis, we will not have the best treatment method. Sorry, excuse me, just a second. Well, so here I can show you this paper. This is based on the United States data. This is in 15 community sites, meaning not specialized hospitals, not university or academic hospitals. And it was in between 2013 and 2015. But as you can see here, it is hopeless to see that only 25% of patients were tested for ROS1. But more important yet, we cannot just, of course, we are now speaking about ROS1, but we will never look only for ROS. We need to look for all these drivers, because all these drivers represent an opportunity for treatment for the patients. So if we look at the last column, we can see that only 8% of patients were tested for all recommended mutations at that time. And these are community, I mean, regular hospitals. So United States. Fortunately, things are moving fast in this aspect, and in recent years, we have improved our performance trying to detect the treatable mutations. But this paper that was published in JCO Precision Oncology this year was comparing what was the yielding of the detection of driver mutations if we use liquid biopsy or tissue biopsy. But what the data I'm showing you is very similar to the one I was showing you before in the United States. This is 2016 and 2017. These are 6 hospitals in the area of Barcelona, but these are university hospitals. These are probably very good hospitals in the management of non-small cell lung cancer. And what we can see here is that, again, and this was a little later, we were only testing 26% for ROS1. So this needs urgently to be improved. And I know that now it's improved. I mean, and probably if we could do this study again, ROS1 testing would be much better, because now we have more options for treatment and more options, more techniques. But again, 0 of the 186 patients were tested for all 8 biomarkers at that point. So we urgently need to improve in this area. Another question I would wonder, I would ask myself is, what caused my cancer? A very difficult question to ask, and probably no doctor will be able to answer you. First of all, it is very important to pinpoint that there is no one case for cancer, not even tobacco, that we know that is the more potent carcinogen related to lung cancer in the world. But it is always a conjunction of factors. Some factors depend on the host, it's meaning myself, my immune system, my age, because cancer is an aging disease, the susceptibility I may have as a population, depending on the country I live or other aspects, and my own gene susceptibility. Also, we have factors that are in the environment, the pollution, anything that I breathe. If the air I breathe is not clean, probably this is damaging my lungs. And also we know radiation, and as Dr. Besse explained, radon, that I will speak a little more. But then is a third part of the pie that it's growing in interest, that it's called microbiome, and the relationship we have with thousands of microorganisms in our bodies, in our lungs, in our gut, in our oral cavity. They are, we are never alone. We all have these microorganisms that are directly interacting with us and with our immune system, and that we know that they can be related. They are related with the development of some kinds of cancers. So as Dr. Besse was explaining, radon is a radioactive gas that is created during the degradation of uranium in the inner part of the Earth, and it filters to the surface and tends to accumulate inside buildings, especially if the places are not well ventilated. And it is the second cause of lung cancer after smoking. So this is very interesting. And what you can see here is that there have been some previous studies that show that the higher the exposure to radon, particularly in this one from Juan Rovina, that is an epidemiologist from Galicia in Spain, the higher incidence of driver mutated lung cancer, particularly you can see there ALK tumors are much more frequent in areas with high exposure to radon. And conversely, the Radon France study showed that areas with low radon, they didn't have oncogenic driver tumors. So probably this environmental radiation creates a propitial status in the lung to the acquisition of mutations. And particularly this study, Radon France, that was presented in 2019 by Laura Mezquita, showed a significant, statistically significant increase in ROS-1 in areas with higher exposure of radon, although of course the number of patients is very low. And as I was saying, the microbiome and its interaction with our body is very important in the aspect of tumorogenesis. In the upper part, you can see the gut, the intestines, where we know that there are layers of bacteria that they live there, and we usually have commensal beneficial bacteria for us that help us to digest and help us to have a healthy immunity. But if there is a dysbiosis, I mean, if the proportion of the bacteria living there changes, that there can appear wealth of substances that can produce inflammation and drive to carcinogenesis. And it is more and more recognized now that lung microbiome is also important in the aspect of lung cancer production. And lastly, I wanted to show you also about the possibility that HPV, human papillomavirus, that is linked with cervical cancer, genitalia, masculine genitalia cancer, and oral cancer can also be involved in lung cancer. So these aspects are to be taken on account. But I'm surprised that we have so little data on the causality or the etiology of lung cancer, and particularly those that are characterized by driver mutations as ROS1. We don't know, and we should know, because if we would have an idea of how they develop, we could prevent them. So another aspect I think it's important is that some of you that the ROS1 non-small cell lung cancer patients may have had thrombosis, because something that is known is that ROS1 positive tumors have a higher proportion of venous thromboembolisms and or any kind of thrombotic events. And sometimes this can be the first symptom of your disease. Repetitive thrombosis, some doctors decide that they need to study why you're having these thrombotic problems, and then they detect the lung cancer. And as you can see here, this is a Taiwanese study, but it's reproduced in many other publications, that the ROS1 patients have a higher risk of thrombotic problems. And the theory, the hypothetical rationale for this is that these tumors, ROS1 tumors, they produce mucins. The tumor cells with ROS1 fusions are very active producing mucins, and these kind of substances interact with platelets, activating P and L selectins and favoring the thrombotic events. So it is very important to know that mucin productive tumors like ROS1 or ALK are more related to thrombotic events. And what is our approach to this? Standard approach is none. I mean, we are not doing anything specific against the risk of increased risk of thrombosis. Of course, if we give a patient a treatment that is working and the tumor is reduced, then the risk of thrombosis seems to reduce as well. But we should take this on account and maybe create a specific protocol to prevent thrombosis in ROS1 patients. What is the best treatment for me? Well, there have been talks about this before, but I like this schedule from Drillon, where this is more or less our strategy right now. We detect a ROS1 tumor, so we have ROS1 TKIs. This is our recommended first-line therapy. And these ROS1 TKIs, well, they have some characteristics. We can have the type 1, that are the ones that we are using, versus the type 2 binding TKIs that can be maybe better, more selective if you can develop them. But none of them are specific for ROS. They all have other activities, and we don't have been able to develop yet very, very selective ROS1 inhibitors. They have different pharmacokinetic characteristics and site distribution, which is important for what we will see later. What happens that all the patients develop resistance at some point to first-line ROS1 TKI inhibitors. And there are different ways of resistance. One of them can be because the drug doesn't reach adequately the intracranial area. So we need better TKIs that reach the intracranial with anticranial activity. Some of them acquire ROS1 resistant mutations that have been described before. Then we need a stronger, more potent, or more specific binding ROS1 TKI inhibitors, next generation inhibitors like lorlatinib or repotrectinib that have been discussed before. But some tumors acquire ROS1 extrinsic resistance, maybe other pathways are activated, so we need combinations. We need to maintain the ROS1, but we need to probably add other drugs. And what happens if we have a mixed with intrinsic and extrinsic resistance? We need to use better next generation ROS1 TKIs and also add new TKIs. Or we have our regular approaches, which are chemotherapy, immunotherapy, or combinations of these with ROS TKI inhibitors that we have not tested yet. So all these areas in blue and a lot of these areas in green and red, we should test. We need to test because this is the strategy. It looks very nice, but we don't have many of these options ready yet in the normal clinic. So as I said, well, this is more or less a summary of what I explained before. it has also been said that pemetrexed-based chemotherapy seems to be more active in patients with ROS1, and it is interesting that it may be related with low levels of thymidylate synthase mRNA in these tumors. So this is something that we could check if we want to treat patients with pemetrexed, for example. Immunotherapy has not been evaluated because most patients with ROS1 were not included in immunotherapy trials, or were included but without knowing that they were ROS1. So we don't have real information, and it will be difficult to collect this kind of information. But something we know is that usually oncogenic-driven tumors have low tumor mutational burden, that it seems to be a particular predictor for good response to immunotherapy. And very similar to what my colleague from the Netherlands showed, we are working with patient-derived cell lines, which we try to understand much better and test the drugs in the lab before you can give to the patients. This is a real patient. It is the pleural fluid. We were able to produce this cell line, and we test different drugs. And as you can see here, this particular cell line that in a patient that was resistant, has derived resistance to crizotinib, was apparently in our cell lines were very sensitive to cisplatin or repotrectinib. Unfortunately, she's responding now to cisplatin. And this is other kind of analysis we can do with the help of also with bioinformatics, as a previous speaker mentioned. We can in these cell lines that were derived from our patient, we can measure the expression levels of more than 700 genes and create these graphs to identify the pathways that are really activated, in red the genes that are activated, and try to target them. So another question I would ask myself if I was a patient is how I deal with the side effects of the drugs. Okay, we have treatments that works more or less. Okay, I'm not cured, but I can stop the disease. I can reduce it for probably a long time or a nice period of time, but I may have some side effects. And what is what do we know about the side effects? Well, there are on-target adverse events. I mean, if we block TRK, we know that any drug that blocks TRK causes ataxia, cognitive changes, etc., as you can see here. If we block MET, we know that we can have peripheral edema, hyperbilirubinemia. These are effects that are related with the target we are blocking. But the problem with ROS1 and also ALK is that we don't know what are the normal function of these targets, these receptors. We don't know what they do in normal adults. So we don't know what are the effects of blocking them. And we don't know because we don't have selective inhibitors. All the drugs we have, as you can see here, crizotinib blocks ALK, but also blocks MET, but also blocks TRK. So all the side effects we see we don't know if are related with ALK blocking or ROS blocking. Okay. Then there are what we call cross-class adverse events. I mean, happening in many types of tyrosine kinase inhibitors, not directly related with ROS or ALK, but with the molecules we use. For example, any oral medication can cause vomiting. This is known, or problems in the intestine when it has to be absorbed. And also there are specific side effects that are related with particular molecules, as lorlatinib, that it causes hypercholesterolemia and hypertriglyceridemia that is not seen with other drugs. But we need to understand the real function of ROS. If we are going to mess up with ROS blocking it, it would be very nice to know what really ROS works for. And this is my last slide. If I was a patient and I would see that I have a rare disease and that the problem is very complicated, I would ask myself, how can we accelerate research? Because research is an urgent need. We need much more knowledge to be able to effectively diagnose and treat my disease. So I don't have answers. I don't have more slides because I would like that the answers will arise in the discussion and in the questions and answer. Thank you very much.
Thank you so much, Dr. Vittori. I think you have put together many questions that certainly raises in the heads of many patients, highlighting the importance of a proper and accurate diagnosis, to know more about the biology of the disease, because we still have a lot of questions, as you said, about how this works and how this needs to be treated. So I think we will have some time to discuss around that on the Q&A session. So thank you so much. So last but not least, I would like to introduce our last speaker, who I have to confess that I'm a very big fan of as a patient advocate. She's Jeanette Freeman-Daly. She's a lung cancer patient and co-founder and board chair of the ROS-1, among many other different things, and a very inspiring voice for all of us working in the patient advocacy community, because of the freshness of the ROS-1 and the many achievements that you have gained, and a truly inspiration for most of us. So Jeanette is gonna talk us about the patient experience. So without further ado, Jeanette, please, the floor is yours. Thank you.
Thank you very much. I'll bring up my slides.
Good.
Okay. So thank you for inviting me to share my story today. I was diagnosed at age 55 with advanced non-small cell lung cancer, which became metastatic despite 2 lines of treatment. And yet here I am still enjoying a good quality of life almost 10 years later, thanks to cancer research and online patient communities. So today I'm going to share my experience of ROS1 cancer and also tell you about the ROS1ers, an international group of patients and caregivers who are living with ROS1 positive cancers of the lung and other organs. In early 2011, I was healthy, a little overweight, and exercising regularly. However, I'd had a light cough for a few months, kind of a My doctor prescribed 2 rounds of antibiotics. I got worse. My doctor then sent me for a chest x-ray and then a chest CT scan. And 2 days later, I was confirmed with lung cancer. I couldn't believe the diagnosis. 2 weeks later, after several tests, I was diagnosed with stage 3A lung cancer, which is considered curable. So I had curative chemo and radiation. My next CT scan showed all the tumors had shrunk or had completely resolved. And the doctors thought they might be able to eliminate my tumors entirely by removing my left lung. I had 16 medical tests in 15 days to determine if my body could tolerate living with just 1 lung. The last test was a PET scan. It showed the cancer had moved into lymph nodes at the base of my neck. I was now borderline stage 4. The shock bowled me over like a 50-foot wave. I went from expecting cancer treatment to cure me to likely not being curable at all. Instead, my cancer grew immediately after treatment. The feeling was overwhelming. Life with lung cancer and on treatment is like being on a roller coaster. Lots of ups and downs. I had to wait a month to start cancer treatment until my obstructive pneumonia was resolved. The day I was supposed to start radiation therapy, I woke up at 3 a.m. coughing up a lot of blood and had to go to the emergency room. Chemo caused my white and red blood cell counts to drop dangerously low. I developed radiation pneumonitis, which is an inflammation in the lungs. And after my second line of chemo and radiation, the cancer started growing again, this time in my other lung. I was now officially a metastatic cancer patient and was not curable. The lifeline for me was a group of people I met in online lung cancer communities. They offered hope. I could see others living well with lung cancer despite a dire prognosis. They offered support. I could connect with others who had the same cancer and were on the same treatments, dealing with the same side effects. I could find someone who understood my experience and feelings because they were also living with it, and they were available at all hours of the day and night. They offered education. Experienced patients were mentoring the newer patients and took time to explain the details of all the data that my doctor dumped on me. I found recommendations for online resources that were trusted. I could track conference findings and new research. They offered empowerment. They helped me find experts in my disease and sources of knowledgeable second opinions, especially useful when one's doctor had never met another patient with my type of cancer. They enabled me to participate as an equal partner in my care team. And they offered connections to others in the lung cancer community, such as expert researchers. In these groups, I learned about a new revolution called precision medicine, which those watching the seminar are probably familiar with. I learned about biomarker testing, genomic tests, and clinical trials. I managed to get my own tissue tested and discovered that I was ROS-1 positive after my second progression. I joined the clinical trial for crizotinib for ROS-1 in November of 2012. And after my very first scan, all my cancer was no longer detectable on scans, and it's been that way ever since. I'm over 8 years, no evidence of disease. This is huge. I went from expecting to be on chemo for the rest of my life and having that life be very short to living a normal life with some side effects, admittedly, but wow, what a difference. This gave me a deep sense of gratitude. I'm not cured, but I'm living well with metastatic lung cancer. And I might be able to stay on this drug for years more. No one can tell how much longer I will be on this drug. I'm an outlier. Being given a second chance at life, though, however long it might be, does tend to give one a different perspective. I have more enjoyment of the natural world. I'm more appreciative of life and its experiences. But something nagged at me. Why? Why was I spared when others died? Why does my lung cancer have an effective treatment when so many others don't? Why am I able to see one of the best lung cancer doctors in the world when some patients can't get diagnosed? Why am I still here? I realized I wanted to help other patients in some way, like the online community had helped me. I wanted to give back. And gradually, I found a new purpose, improving outcomes for lung cancer. Everyone in this picture is living with lung cancer. Some are now close friends. Many are on clinical trials. Some have passed. Some have done everything right. Some have done things that affected their health. But that doesn't matter. No one deserves lung cancer. In 2015, a group of like-minded patients met up. All of us had ROS-1 positive cancer, and we all wanted to do something. The ROS Wonders was born. The ROS Wonders is a global group of patients and caregivers dealing with ROS-1 positive cancers of all types. We're the largest group of ROS-1 patients ever gathered. There's over 670 in our Facebook group. We strive to improve outcomes for all ROS-1 cancers by supporting patients and caregivers, increasing awareness and education, accelerating research, and improving access to effective treatment and diagnosis. Patients and their caregivers meet in a private Facebook group to share experiences and information about cancer treatments, side effects, living well, and sometimes dying. We also share educational information vetted by ROS-1 researchers and clinicians, and sources for ROS-1 second opinions. We share research updates, changes in care guidelines, and other news. To promote trust and transparency, we emphasize citing reliable sources when we share medical information. Nobody gives medical advice, but we can all share our experiences. And we partner with advocacy organizations, clinicians, researchers, and industry to create research projects. We're truly a global collaboration. We have patients currently in 30 countries. At times it's been as high as 36. And well, this is 8 cancers, but there were 12 different ROS1 cancers that are known. We have 8 in the group. We also have groups that have formed in other countries as people want to gather and speak in their own language. So there are groups in Australia, the Netherlands, China, Spain, Italy, Germany, the UK, Japan, and France. People who are members of these other language groups can come into the English-speaking group to find information, and then they take it back and translate it for other people in their own groups. As a group, we've initiated several projects and get involved in a variety of things to help drive research. We have webinars with experts and Zoom meetups where patients can talk to each other. We attend conference presentations and have made several ourselves. We network with a variety of researchers all over the world to try and find the best information we can for our disease. As some of the other people have mentioned, we started the ROS-1 Cancer Model Project. When this began in 2017, there were just a handful of ROS1 cell lines available. Through our work with the University of Colorado, we've helped to create 9 new cell lines and characterize them. And they've been shared with other institutions to try and develop new biomarker tests to look at acquired resistance. Some of the presentations that you saw earlier showed work that was based on our cell lines. And also look at new drug treatments that we might have. We just last fall had a ROS1 research roundtable where we brought together ROS1 researchers and clinicians from across the globe to talk about what we could do to help accelerate research, and generated a white paper that has come up with suggestions for what we can do. One of the things we're doing is to try and take our cell lines and the characterization data and make that information widely available so that other researchers can build on our work. We're also looking at developing a collection of what they call real world data of what it's like as the lived experience in ROS1 to see if we can find more information that will help us. There's also a variety of research going on driven by ROS1ers in other countries. There's a grant in Belgium that's working on ROS1 cell lines and using CRISPR technology. You've heard Vall being spoken about in the Netherlands. There's a project under development called Uncover ROS1 in Germany. And Australia has created a ROS1 registry. There are ROS1 members involved in all of these projects. So everyone has their own why of why they want to get involved in advocacy or in something like the ROS1ers. Mine are my family, the growing lung cancer community, in which I've made some lifelong friends, and improving outcomes for ROS1 cancer patients. I'm a full-time research advocate, and it's my passion. That's what keeps me going, even when I feel overwhelmed by symptoms of treatment or side effects, or just worrying about when the other shoe is going to drop and my cancer might progress. My story shows that you can live well with metastatic ROS1 cancer. It encourages patients to ask questions about their treatment. It helps family members of cancer patients understand their loved ones better. It helps doctors realize that patients can be partners in their own care. And the ROS1ers are showing advocacy groups, clinicians, researchers, and industry what true patient-partnered research can look like and accomplish. I live with the downsides of cancer because I hold on to what matters most. While precision medicine has allowed me to live with cancer as a chronic illness, it's not the same as the life I had before cancer. I want to see that change. I want everyone to have the benefits of good cancer treatment. I want everyone to be able to get tested for all of the appropriate biomarkers. The path forward for us is more research. And because ROS1 is so rare, we can't do research by ourselves in any one country. We really do have to work together. There are a lot of barriers to working together on international research, but I'm confident that if we all work at it and join forces, that we can help to accelerate ROS1 research and find new treatments and better cancer care for all of us. Thanks for listening.
Thank you. Thank you so much, Janet, for this to listen. I know that many of the people in the session today will be very familiar with your testimony, and they will find very identified with you. And thank you for all the work that you're doing for the community that shows that patients can also play an active role in things to collaborate and to improve and to participate actively in improving research and improving outcomes for the community. So thank you so much. So right now we are moving forward to the questions and answer session. I would like to ask all the panelists, all our speakers today to come up, to enable your cameras, to open up your microphones. I have to say that we have like around 50 minutes. We have received a good bunch of questions, so I'm not sure whether we could cover all. So what I'm going to do is I'm going to start asking questions to some of you, but please, any of the speakers, feel free to adapt to the questions that our audience has sent us. So the first one I would like to ask Dr. Felip, a question from one of our attendees today, who says, would you recommend to all patients at least to ask their oncologist to get an NGS analysis even after being diagnosed by FISH as ROS1 or ALK positive?
Thank you for the question. Excellent question. I think there is no need to perform NGS if we have a diagnosis of ROS positive by FISH. My presentation was more in general about the need to perform NGS probably in all patients at diagnosis in order to diagnose all potential alterations. But if there is a diagnosis of ROS by FISH, there is no need to repeat an NGS. Thank you for the question.
Thank you.
I would like to just add something to that. We have had some cases where someone has tested positive for ROS1 and then did not respond to a ROS1 targeted drug. And in that case, you would want to get a different form of testing to confirm you really do have ROS1.
I absolutely agree. The response, as we discussed, the response rate in ROS1 is really high, with crizotinib and agents. If we have something that is different of what we are expecting, of course, we can do. My answer was more in general. I think for if we have a diagnosis of ROS by FISH, we can start the treatment, and I think that's it.
Yes.
Thank you.
Thanks for that. So I'm moving to the next question. This was sent to Dr. Besse, but as I said, everyone feel happy to join because I think it's a very interesting question, which says, how do you think patients can help and campaigns to influence regulators and payers to be flexible with the drug approval and access criteria?
Well, I'm probably Janet might have some ideas on that. I know that some drugs have been refunded because the patients community discuss with the payers and explain to the payers that it was the only option they had. And I'm completely amazed by the level of efficacy that is given to the ROS1 drug in my country. It's completely disconnected of the reality. The health authorities ask us to. Sorry, I am my cat.
I know that feeling. This is next to. The health authority asked us to do a randomized trial, and all the academic society in France explained that it was not feasible in 1% of lung cancer patients to run a randomized trial, and still there is struggle to get a refunded. But maybe, Janet, you have some ideas.
Yes, it is a problem when there's so few patients. It's very difficult to run a phase 3 trial to compare two drugs. Some of what the ROS1ders have done and some other organizations have done is For instance, in the UK, when crizotinib was being evaluated, we gathered input from various patients who had been on crizotinib and used that to demonstrate that these patients who would normally be given less than a year to live were still alive years later, had a good quality of life, were able to continue working and paying taxes, taking care of family members and contributing to society. And that even though they might not be cured, it still made a significant difference. It doesn't automatically get the drug approved, but it did allow crizotinib to go on their experimental drug list. Canada did something similar and was able to get crizotinib approved in their country. And the Australian patients also submitted information that helped get drug approved there. So gathering real world data and sharing it with regulators is the best option we have, I think.
Thanks a lot for that. And yes,
Can I add something?
Yeah, for sure.
Yeah, I think, well, in addition, I think for Europe, it's really a problem that in every country, you have different regulations. And well, even the fact that you have one EMA which approves some drugs, then still you have these different items, what happens in different countries. So I think that the patients of the different countries in the EU have to unite and talk with their doctors how they can help in their country, I think. So to join in all the countries to get the right drugs approved and reimbursed, I think.
Yeah, Dr. Viteri, you might want to add something, yeah?
Yes, I was thinking that maybe we should learn some lesson about the COVID pandemic, no? Where there was lightning fast approval of vaccines, clinical trials that were incredibly accelerated by more flexible standards. So this was because it was an urgent issue of worldwide magnitude. But of course, for a patient that is diagnosed with cancer, it's of urgent need to advance. So I think that now we have learned that we can do things fast when it's needed. So we should learn that lesson and continue applying it for many other aspects.
Thank you so much. And just to add on, as I'm participating on behalf of Lung Cancer Europe, this European umbrella of patient advocacy organizations, when it comes to the regulatory side, we try to influence at the European, or participate in the in some of the committees that the European Medicines Agency has in place. But when it comes to access in Europe, this is something very nation related. So for those of you attending who are involved in patient advocacy groups, you should really explore the ways in which how you can participate, especially with an evidence-based model to really put the patient needs on the table of payers. I know, for example, in Spain, there are some mechanisms of participating in health technology assessment process. There might be limited, but there are some ways. So if you are interested, please be in touch with us so we can help you. Okay, so I'm moving to the next question. This one is to Dr. Pauwels, because we have a patient who is also as well a bioinformatician. And he's asking us for the use of machine learning. A lot of data is needed to train the models. So do you think today there is enough data to do that? And if so, would you recommend where we could get that data from? Yeah, Dr. Paul, I think, yeah.
This is, yes. So this is a terrible question for asking a pathologist about that. You know, Geert van der Weijer is a bioinformatician, and his wife is ROS-1 positive. And now he is making a crusade for ROS-1 in bioinformatics. So I would leave this question to him, but he is not here, I suppose. I cannot save myself. But indeed, data are coming up. Remember, 10 years ago, we only tested for one gene at a time. Now with NGS sequencing, we are moving to more and more genes, but we are moving also to more and more data. So if you have a small, very good, but small panel for your NGS containing 30 genes where the actionable mutations are, that's very fine and it works very well. But now we come with panels of 400, 500 genes, even 1,000 genes, and it makes my life and your life more complicated because we don't see only the driver mutation, but all other mutations that are happening. And this is what I wanted to share with you with my slide about how simplistic we are thinking about pathways. Remember EGFR, KRAS, BRAF, MEK, ERK, whatever. The reality is far more complex, and that's where bioinformatics comes in. Only speaking about mutations, if we have a list of 20 mutations on the ROS1 fusion, well, these are data that you have to collect, and then you have to use, indeed, as has been mentioned, artificial intelligence to make something more meaningful about this data collection. And we are moving to that. But this was an example of mutations. But we, as I said, we will have data on proteomics, on epigenetics changes in these tumors and so forth. So I think your ROS1 bioinformatics patient has a lot of work to do.
And
maybe in addition, I think we need lots of material. So we need material from many patients. That's what I was talking about, of course. And I think that when we have more material, we have to work together. So I will speak with Geert again, hopefully in a short time, but try to go on with it. Yeah.
Another aspect that we're looking at is perhaps just looking only at ROS1 might not help us as much, since half of our patients develop resistance mutations on pathways we don't know anything about. So part of what came out in our white paper is we need to start looking at resistance that develops in these pathways across these rare fusion oncogenes and see if we can build more data that way by looking at more than just one type of gene alteration.
Great. I think we still may have time for a couple of more questions before closing the session. So this one, I mean, was targeted to Dr. Viteri, but I think everyone can answer here. So because Dr. Viteri, you spoke about the lack of knowledge of some oncologists about ROS1 because they don't see many ROS1 patients. So how can patients know if the doctor is familiar with this mutation? And if you go to the patient, if you go to the doctor, you know nothing at the beginning as a patient, you lack the information, and you know nothing about molecular profiling. So what should patients take into account when visiting a doctor which may have no many experience in treating this type of mutations?
Well, there is one thing that the oncologists usually are very industrious people that like to study a lot. So they may have not seen a patient in real life, but they may have read a lot of articles and papers regarding the treatment, the side effects, etc. Fortunately, regarding ROS1, the treatments we use are similar to ones we use in ALK, which is a little more frequent. So probably it's not so completely unknown territory, no? But the truth is that oncology is vast and difficult to comprehend. And so what is happening in modern oncology is that we are subspecializing, and many of us, well, we don't see breast and lung and gastric cancer because it would be very difficult to be aware of anything that's relevant in this area. So, but unfortunately, in the model of Spain, at least, there are many little hospitals that have oncology departments, which are useful for many aspects, but are not useful for very uncommon diseases. I mean, if you have a lymphoma, you know that you cannot be treated in any hospital. You must be directed to a particular kind of hospital, a referral hospital, etc. In this case, I think some particular kinds of non-small cell lung cancer are similar. I mean, you should like to ask your oncologist if she or he are specialized on lung cancer. This would be a good start. And ask them if there are clinical trials that he or she are aware of, which is always an important part of the treatment. So to start this would be very good points, to know if your oncologist is specialized in lung cancer and to know if they have connections with clinical trials.
If I may also add something, we have a page on the ROS1cancer.com website where we list the people we know have experience with many ROS1 patients, and we're in the process of updating it. So while many researchers or many clinicians can look at the guidelines and choose a first-line drug, it gets more difficult once the patient develops progression, and it might be useful to get a second opinion from someone who's dealt with a lot of ROS1 patients.
Thank you, Jeanette. And I think we have time for a very, very short question. And this is directed mainly to Jeanette because of your experience, but any other tips may help. So, you know, one of this meeting has been possible because a small group of patients across Europe connecting through Facebook and WhatsApp groups wanted to do something in Europe and start contributing to the community and so on. And it was challenging because most of them have been diagnosed, but they didn't know exactly where to start from. They wanted to come together and hopefully, thanks to this meeting, there will be a good bunch of European patients connected. And this could be, as in the movie, the start of a long friendship, let's say. So from your experience, what would be your tip or your advice? Where to start from? What could be the first low-hanging fruits? Yeah.
I think working with the groups such as Lung Cancer Europe and some of the others that work across the countries would be a good place to start. As I understand it from talking with our European ROS1ers who are on our drivers, the committee of leadership, the barrier is language in a lot of cases. Every country has different regulatory system, they have different healthcare system, different drug approvals, but trying to create something that can be translated by everybody, even if you just choose a language that's predominant in a country, you may not cover all the dialects. I think being able to tackle the translation issue is big. I know that our ROS1ers such as Meryl in the Netherlands, Geert in Belgium, and Berbel in Germany have been working on trying to pull together the European groups and try and see what they can do together. So I would think talking with the people who've been working on this for a while and talking with the agencies that are working on translation would be a good start.
Great. So it seems suddenly there are a lot of other questions, but certainly we have run out of time. And before closing, I would really like to thank first of all of you, the speakers, for sharing with us your valuable time today. I think it's been, for me, it has been an honor to be able to chair or to moderate this session. I think what you have shared with everyone has been truly great. I would also like to thank the Spanish group for people living with lung cancer, IACAP. I can see now Bernard on the screen for giving me the opportunity today to be here. It's been a real pleasure. Also, I would like to thank the sponsor who made possible this streaming, ROS, Takeda, and Pfizer. And of course, to all of you who attended here today. As I said, I think this is a great opportunity to connect you all and start doing things. So thank you very much for joining. So from my side, I just would like to wish you all a very good rest of the evening. I'm not sure, Bernard, if you would like to add some minor words to end up the session, but from my side, thank you very much all for joining and have a very good evening. Bernard, would you like to add up something before we close?
Thank you for all. I hope that the patients, English is very bad, I'm sorry.
No problem. I think the good thing, as Janet mentioned, and something that we usually say in the European patient advocacy community, is that the official language is bad English. So no problem at all. Okay. So thank you all for being with us today, and I really wish you a very fruitful evening to everybody. Thank you so much. We'll stay in touch.
Thank you for organizing.
Adios.
Thank you.
Bye-bye. Okay.